Pharmacodynamics refers to the mechanisms and effects of medications within the body. Or more simply, it's what medications do to the body and how they do it. Alright, so in order to have an effect, most medications have to reach their target cells and bind to a receptor. Receptors are specialized proteins both on the cell membrane and inside the cell that can bind to a ligand and get triggered to alter their shape or activity. This gives rise to a signal cascade of intracellular molecules, which are the second messengers, which ultimately results in some change in the cell's function. Intracellular receptors are typically found in the cytoplasm or nucleus of the cell, and recognize small hydrophobic ligands, or water-hating ligands. These include molecules like steroids, which are happy to diffuse across the phospholipid membrane. Once bound to their ligand, the receptor-ligand complex attaches to specific DNA sequences that activate or inhibit specific genes. On the cell membrane are cell surface receptors, which are embedded into the plasma membrane and bind to ligands too large or hydrophilic to pass through. Hydrophilic meaning water-loving. Based on their structure and properties, cell surface receptors fall into three main types. First are ligand-gated ion channels, which form channels or pores that are generally closed, but then open up once they bind a specific ligand. They allow ions like chloride, calcium, sodium, and potassium to passively flow into the cell, down their gradient, and trigger the signaling pathway. Next are the G-protein coupled receptors, also known as 7-pass transmembrane receptors, which means they're really long proteins that have one end that sits outside the cell, and then the snake-like protein dips in and out of the cell membrane 7 times, and finally ends on the inside of the cell. The ligand binds to the end sitting outside the cell, and the end of the protein that's within the cell activates guanine nucleotide binding proteins, or G proteins, which contain an alpha, beta, and gamma subunit. Normally, the alpha subunit binds to guanosine diphosphate, or a GDP molecule, and the G protein is inactive. When a ligand binds to the receptor, the G protein changes shape, causing the alpha subunit to release the GDP and allowing a guanosine triphosphate, or GTP, to bind. This causes the alpha subunit to detach and trigger other proteins in the signaling pathway. Broadly speaking, there are 3 types of G proteins: GQ, GI, and GS. Each type has its own kind of alpha subunit. The GQ protein activates the enzyme phospholipase C, which cleaves a phospholipid called phosphatidylinositol 4,5-bisphosphate into inositol triphosphate and diacylglycerol. On the other hand, protein GS and GI stimulate or inhibit, respectively, the enzyme adenylate cyclase, which takes adenosine triphosphate, or ATP, and removes 2 phosphate molecules and transforms it into cyclic adenosine monophosphate, or CAMP. Inositol triphosphate, diacylglycerol, and CAMP then go on to stimulate and inhibit different sets of enzymes and molecular pathways. Finally, there are enzyme-coupled receptors. These are usually single-pass transmembrane proteins, meaning that they only have one transmembrane segment. The extracellular end of these receptors bind to ligands, and their intracellular end has enzyme activity. The enzymatic domain is usually a protein kinase, which phosphorylates other molecules. When a ligand binds, it triggers a conformational change in the enzymatic domain to form high-affinity binding sites for the second messengers. These second messengers get phosphorylated by the tyrosine kinases before heading off to activate other proteins in the signal pathway. Alright, so every cell has a unique combination of many different types of receptors. And in theory, each medication is specific to a particular receptor, just like each key fits into and opens a particular lock. This is described as a medication's specificity. In reality, though, no drug is 100% specific, meaning it could also bind to other receptors that are similar to its intended target, which is why all medications cause side effects. Now, the way a medication and receptor interact with each other is ruled by 2 main principles. Affinity is how strongly a medication binds to its receptor. This is mainly determined by the strength of the chemical bond between the two. The higher the affinity, the higher the potency, which is the amount of medication needed to elicit an effect. So medications with high affinity can produce an effect at a lower dose. After the medication binds to its receptor, it must also be able to activate that receptor. This ability is known as intrinsic activity. Ultimately, the intensity of response to the medication depends on both the number of receptors bound to the medication as well as its intrinsic activity. The maximal effect the medication can produce is called the efficacy. Alright, now let's plot all this into a nice graph to show the relationship between the concentration of a medication in the body, which depends on the amount of medication given, also known as the dose on the x-axis, usually on a logarithmic scale, with the response produced on the y-axis. What we get is an S-shaped curve, called the dose-response curve. At first the curve is more or less flat, and that's because the concentration is too low. So not enough receptors to bind to the medication to cause a significant response. As the concentration increases, more receptor medication complexes form, so the response to the medication climbs upward. This is described as a graded dose-response relationship. Eventually, we reach a point where all of the receptors get occupied, so the curve starts to flatten out. This is where the maximum response or effect, abbreviated as Emax, is achieved, which is a measure of the medication's efficacy. Now, if we move to a point where 50% of the maximum effect is produced, the effective concentration of the medication producing this effect, known as EC50, marks its potency. So the smaller the EC50, the less it takes to get halfway of the maximum effect, so the more potent that medication is. A similar concept to that is ED50, which is the effective dose of a medication that needs to be given to a certain population so that 50% or half of them see an effect. To illustrate this, let's plot the dose-response curves of two medications, medication A and medication B, on the same graph. Notice that both reach the same maximum effect on the y-axis, so the same efficacy. However, on the x-axis, the EC50 required to produce 50% of that effect is much lower for medication A than B, meaning medication A is a lot more potent than B. In other words, the closer the curve is to the left, the higher the potency is. Now let's look at two other medications, C and D. Medication C achieves a higher maximum effect on the y-axis, meaning that it's more efficacious than D. In other words, the higher the curve, the higher the efficacy. At the same time, though, on the x-axis, the EC50 for 50% of that effect is the same for both, so they're equally potent. Alright, as a quick recap. Medications produce their effects by interacting with receptors, either intracellular or extracellular ones, including ligand-gated ion channels, G-protein-linked receptors, and enzyme-linked receptors. These interactions are characterized by the medication's specificity for the receptor, affinity or strong binding with the receptor, and intrinsic activity, or ability to activate it, which in turn determine its potency, or the dose needed to produce an effect, and efficacy, or the maximum effect the medication can produce.